Hi everyone, here we are in video lecture number 3 of microbial control, and we just finished up physical methods of control, and there was a really nice summary in your textbook. Reading your textbook on this whole unit is really helpful because it has, it will just help solidify this information in your mind, and I highly recommend going back in and reading it. Um, look over this for reviewing and for studying for tests and quizzes. So um, I think it's a really nice table right there. The right after that, the next topic I have is describe how surfactants are used to control microbial growth. And I would consider this a physical method of control, but it's also kind of a chemical method. What happens with surfactants, and when we talk about surfactants, we're talking about things like soap. And what soaps do is they break up oils. So they have a chemical function where they can kind of break up the connection between microbes and oils on your skin. And that's why washing your skin a lot with soap can dry it out because it breaks up the oils. And then what we use is water to rinse that off. So kind of the mechanical action is between the scrubbing and the rinsing off after you break the oils. It's kind of a physical thing, but you need the chemical action of the soap breaking up the oil molecules to be able to then rinse off the microbes mechanically. Now, soap itself or surfactants themselves do not kill microbes. And, so I think most people think about that. Oh, I washed my hands and I killed all the microorganisms. But what's the important thing is the rinsing of getting removing the microbes off your hands. And it works really well. This should look familiar from our hand washing experiment. And so this is at one of those Savarod plates where we had yeast on it. And it's showing you that, unwashed hands with this transient on it, we can see lots of colony growth. And then the more we wash our hands, the more you're going to remove those colonies, remove those yeast cells. But, look at that. It's not 100%. And that's important to remember. Even though you wash your hands, there's potential that you're still carrying some transient organisms that could be potential pathogens. That being said, if you tried to count the colonies in one of these fingerprints, I don't know how many would be in there. Maybe you could get to 100 or something. But if I count the colonies in one of these fingerprints, I have less than 10. And so you reduce it at least, I would say definitely you'd get it down to 90% of those transients had just washed off. So hand washing really is one of our most important methods of microbial control. And in your textbook, it goes over some of the CDC recommendations for effective hand washing. So you can look at that. What I would recommend doing right now is going to your lecture study guide and doing the matching that we have here. And what you can do is turn off this video, go do that, and come back and get the answers. And so I have the answers, right? So stop this before you see the answers. But I thought I had the answers. Let me see. Sometimes it doesn't advance very well. There we go. There's the answers. So anyway, if you have any questions about those, we can talk about them when we come back to class. All right. So now we're fully into the chemical control of microbial growth. And one of the first chemicals we're going to talk about is phenol and phenolics. And how are they used to control microbial growth? And this photograph or this painting should look a little familiar, similar to that photograph we had earlier in an earlier lecture talking about Joseph Lister during the golden age of microbiology, who started using phenol, kind of sprayed it in the air, sprayed it on the patient, sprayed it on their own hands. Oh, here they're using ether. I don't know, maybe they're ether or chloroform. I don't know which one they would be using at that time. But there's the anesthesiologist, and they're cutting this guy's leg off, or obviously doing some really serious kind of surgery, maybe I don't know if they're cutting his leg off or if they're fixing a compound fracture. But remember, Lister used phenol as one of our first really effective microbial control agents in surgery, and it made a dramatic impact. It really significantly reduced the incidence of infections. Thinking about that, compared to using nothing on your hands to reduce infections, using nothing on your tools or the patient's skin to reduce infections. So, I mean, here they're in a room and they even have a window that presumably is open to the outdoors. The amount of microorganisms around were probably significant. So using phenol was a really important step in microbial control. That being said, phenol itself is a really potent chemical. I've used phenol in research laboratories, and if you use concentrated phenol and you get a drop on your skin, it turns your skin to this like gummy mush. I mean, it's really potent. And so they use dilutions of phenol, but it's still like a strong chemical. You might be used to phenol if you ever used one of those throat lozenges that numb your throat. That's a really low concentration of phenol that in some of those throat lozenges. But we don't use it that much for microbial control anymore because we just have better things. If you look right here, you don't have to memorize this, and it's in your book, but this should look familiar if you took chemistry. You have a benzene ring right here and an OH. That's a phenol um, molecule. That's what phenol looks like. But there's this other group of compounds called phenolics. And so chemists got their hands on phenol, and then they did different chemical modifications to it. And so these different chemicals are phenolics. And so the effort was to make phenol a little less toxic or maybe to last on surfaces longer. And so you'll see phenolic compounds, particularly in some of these original formulations of Lysol. And a lot of the Lysol right now are no longer phenolics, but I mean, probably most of them. But the original Lysol was a really strong smelling product, and it did use a phenolic compound. Other places that you see phenolics are pine cleaners. We used to see them in the store a lot of times that things like Pine Sol, and they would take the pine oil, and that turned out it was a phenolic compound, and it was antimicrobial. Now, again, it was just cheaper to make these synthetic pine cleaners where they put in like chemically synthesized pine cleaners rather than the ones that you would actually isolate from pine trees. But you can still buy some things like that. But they probably have today's may or may not even have some of the phenolic in there, but the original pine cleaners were like that. Some people will say use some of these natural oils like clove oils for antimicrobials, and it's because they also have phenolic compounds in them. So phenolics are good antimicrobials. They're just not used today as much as they used to be. There is a group of phenolic compounds, though, that made a real surge um, in in their use, and I'm sure you remember these. These are all these antimicrobial products that came on the market. I don't know exactly when they came on the market, maybe the 90s, around there, 1990s, and we had all these different products that had what we know to be triclosan. Triclosan is a phenolic compound. This O there, if you take an OH and you attach this on it, or there's another OH. So there's the benzene ring and the OH. It's a phenolic compound, and it turned out that when people made this stuff called triclosan, it had a couple really good qualities. First of all, it was a really good antimicrobial. It was fairly gentle, so you could use it on your skin, and it was really shelf stable. And if you don't, you think about a lot of the things that are in the grocery store, one of the key things to selling products is that you can put it on the shelf and it will last for a long time. And so this antimicrobial was really shelf stable. It was something that you could put it, people put it in all sorts of things. You can see all these antimicrobial soaps, toothpaste. Here they took like toothbrushes, and you could melt the plastic. You'd have hot molten plastic. You could put triclosan in it and cool it, and make a a toothbrush, and the triclosan was still stable. So you could use lots of products with this. If you go to the grocery store now, you're not going to find soaps, hand soaps and whatnot with triclosan in them because, oh, it's been, how long has it been now? Maybe, I don't know, has it been 10 years already that we decided that triclosan was not a safe product to use? And what was happening is because it was so stable and useful in our antimicrobial products, it also was stable in the environment. So anywhere you go into waterways, in natural waterways, you can still find triclosan in there because it's so stable that microorganisms don't break it down in natural environments. The other thing we were finding is there were some microorganisms becoming resistant to triclosan. And so here everybody's using it as hand soaps. Well, it turned out hand soaps alone remove, 90% or more of microbes, of transient microbes. So adding triclosan to it didn't really give us a big benefit. We really didn't need to be slathering our bodies with this. All of you have heard of staph infections. Turns out Staphylococcus aureus, which is the main pathogen in the genus Staphylococcus, is really sensitive to triclosan. So when somebody gets a bad staph infection, it's really helpful to be able to use an antimicrobial soap on that infection. If it becomes resistant to triclosan because so many people are using it all the time, it's not going to be helpful. So the CDC, I think the World Health Organization also got involved in it and said this is just not a product we should be using all the time. So we've got a lot of restrictions on how we use these antimicrobial products. And so triclosan was officially banned in hand soap. So if you're looking for an antimicrobial hand soap now, you'll look at the active ingredients and it's not going to have triclosan. It will have something else. That being said, we still do use it because it's a really effective chemical. I've heard of people going in for surgery. So maybe they're going to get, surgery where there's going to have a large incision and they're told to wash in antimicrobial soap with triclosan in it. They'll get it from a prescription and they'll use it prior to the surgery to reduce the number of pathogens on their bodies. So it can be still a very useful product. So the question right here is it's an exceptionally stable antimicrobial, and that's good because you can put it in a lot of products and it's shelf stable, but it can also accumulate in our environment. I mean, you can find triclosan in people's bodies. And is that a problem? We're not really sure. Accumulating a chemical in the environment that does not degrade, it's always something we should be cautious of. Our next chemical we're going to talk about are alcohols. And alcohols are used for microbial control. I mean, probably everybody in here has used alcohol at some point. You get a cut and you might put some alcohol on it. Oh, I know we've all used it with hand sanitizers. So alcohol is a really effective antimicrobial. I like this table. It's not in your book, but it's something that I've used from another book, but I think it's an interesting point to be made. It's showing you here ethanol is a really common alcohol. It's the kind of alcohol that we drink, and it's showing you 40 to 100% ethanol, and this is saying biocidal action against strep pyogenes. That's the streptococcus that causes strep throat. So what they're showing you is if you take 40% alcohol and you expose strep pyogenes to this alcohol for 10 minutes, the bacteria are still going to be able to grow. And it looks like 40% alcohol, no matter how long you go, up to 50 minutes, it's still going to be able to grow. But as you increase the concentration of the alcohol, it's not able to grow. 10 minutes exposure to 60% alcohol, strep pyogenes no longer will grow. You'll get no growth. But the interesting thing is if you look up here and you get up to 100% alcohol for 10 minutes, strep pyogenes can grow. You treat strep pyogenes in alcohol for 10 minutes, put it on some nutrient agar, it will grow. And it turns out, for whatever reason, this higher concentration of alcohol is not effective. And so many times we're saying, oh, the more concentrated the chemical is, the better it's going to work. But in reality, it's really important to read labels and use your chemicals in concentrations that are recommended. So alcohol is kind of an oddball in that way. Um, Another thing that we use alcohol for a lot is degerming. And you guys are familiar, we're all familiar with this because we've all gotten vaccines. And alcohol is really great at breaking up oils, just like surfactants, like soap. And so what you do is you get alcohol and you put it on the skin, and then you kind of rub the swab in a circle. And what you're doing is you're pushing the oils and along with them the microbes away from the site where you're actually going to give the vaccine. So it's not, the alcohol actually isn't even on the skin long enough to kill the microbes. It's just breaking up the oils chemically and pushing them away from the injection site. So we call that degerming, when you're pushing microbes away. Right here, it's telling you that alcohol can be bactericidal, fungicidal, and even kills some viruses, being virucidal. So, and it just depends on what concentration and how long, but it can be used that way. One of the things to keep in mind is it evaporates very quickly, so it might not be in contact with the surface long enough to kill things, which is why these a lot of these alcohol antiseptics are, the hand sanitizers have kind of a thickener in it so the alcohol doesn't evaporate so quickly. Not effective against endospores or fungal spores. So that'll tell you that alcohol is not very effective at sterilizing. Let's see, let's go here to the halogens. And another chemical, and you might remember this from chemistry, that the halogens are group 7 on the periodic table. Fluorine, chlorine, bromine, and iodine are the main ones that we're interested in for microbial control. And they're very effective at microbial control. The halogens, group 7. And you can probably give examples of how they're used. Iodine is probably one of the oldest microbial control agents we have. And just like when we used phenol, chemists got their hands on iodine and did some chemical variations and made iodophores. And so there's all sorts of different iodine products. One of the common iodine products for we didn't really use it when I was a kid, but before I was a kid, or maybe when I was really young, a lot of people had tincture of iodine in their cabinet. And a tincture is a chemical mixed with alcohol. And it turns out alcohol is a good antiseptic, iodine's a good antiseptic, but together they're they function even better than each added up together. They actually enhance the function of each. So that was a really common thing that people would have in their medicine cabinets. But you can see right here. Oh, and even remember when we used iodine in the gram stain and the starch test, it's got that kind of brownish yellow color. And so it stains the skin. And iodine kind of went out of fashion because of that. Also, some people are allergic to iodine. Probably the most common form of iodine you're going to see in microbial control in your life today is betadine. And betadine, there's a scrub, there's an antiseptic that is available, and it's got iodine in it, and that's the active ingredient. And one of the nice things about the formulation of betadine is it continues killing microorganisms while it's in contact with the skin. So if you swab the skin, you're going to do surgery there or do a needle puncture or something like that. As long as that betadine's there, it's going to continue to kill microbes. If you put alcohol on that area, the alcohol will kill microbes, dry off, and then whatever lands there or whatever continues to grow will be able to do that. So betadine's real commonly used when donating blood also. So it's a really common iodine containing product, or a common halogen. Let's see. Another halogen that we use for microbial control is bleach, and the active ingredient in bleach is hypochlorous acid, which gives us that chlorine. So bleach, the active ingredient in it, is a chlorine product that's a very effective microbial control. This is a handy thing to know if you are in a pinch and you need to decontaminate water from microbial growth. One thing you can do is you can take 2 drops of bleach in a liter of water, and a liter is about a quart, or you know what a 2-liter bottle of soda looks like. 2 drops of bleach in a liter of water, and it's very effective at controlling microbial growth. 10% bleach can actually sterilize. So you can use bleach to kill endospores. This is one of those products at higher concentrations you're not going to get any better effect than sterilization of 10% bleach. You never need a stronger mixture than that. The other thing about chlorine, it's functioning in a liquid form as a form of bleach, hypochlorous acid or sodium hypochlorite. It can also be used as a gas. There, you can use gaseous chlorine to sterilize. Now you can imagine that that's going to be really dangerous. So it's only used in very highly controlled situations where you might need to get into all the nooks and crannies of something. The situation where I know chlorine gas was used for microbial control was, I thought it was like in 2008, 2009, somewhere around there. There were these, somebody was sending anthrax spores through the mail, and anthrax makes endospores. And somebody was powdering them, making weapons grade anthrax, powdering them and putting them in envelopes. You open the envelope and the dust just floated out, and you could breathe that in or get it in a wound and you could get anthrax. So that was happened a few times in the US. And there was a couple buildings, one in New Jersey, one in Washington, D.C., that got these anthrax spores in them. One was a government building and both were government buildings. One was a post office. And a lot of people got sick, and it turned out, how do you clean this? Endospores could last there for 100 years. So how do we clean every nook and cranny? Well, what they did is they tented sections of the building and pumped in chlorine gas and let it sit there to kill the endospores, and then they pumped it out. So it is a useful mechanism to reduce microbial control, to reduce the number of microbes and even sterilize. You're probably familiar with using chlorine in water. So again, it's along the scale of this. In order to reduce the number of microbes. So a little bit can be really effective. I have a little video here. I'm not going to click on it, but if you go to YouTube, there's the address, and click Disney and bromine. It talks about the halogen named bromine that you can also use to decontaminate water. And some of you might use chlorine in your swimming pool water. Other people might use bromine that doesn't evaporate as easily. And there's a really interesting little YouTube video right there that you can go to and go to YouTube, Google Disney and bromine, and you'll see this little video about how bromine is used in the water at Disney. I think that's a good place to stop. We'll go on to the next topic in the next video.